are biological catalysts that contain protein molecules through nature. They are formed by living cells, animal, plant, and microorganism, and are essential as catalysts in biochemical reactions. In a cell, almost every reaction requires the presence of a specific enzyme. A significant function of enzymes in a living system is to catalyze the making and breaking of chemical bonds. Therefore, like any other catalyst, they increase the reaction rate without undergoing permanent chemical changes. The catalytic ability of enzymes happens due to their particular protein structure. A specific chemical reaction is catalyzed on a small portion of an enzyme surface. Enzyme reactions are different from chemical reactions. An enzyme catalyst is particular and catalyzes only one or a small number of chemical reactions. A great variety of enzymes exist, which can catalyze an extensive range of responses. The rate of an enzyme-catalyzed reaction is usually much faster than that of the same response when directed by other types of catalysts. The reaction conditions, temperature, pressure, pH, and so on, for the enzyme reactions are very mild. Enzymes are comparatively sensitive or unstable molecules and require care in their use. Industrial applications of enzymes. Industrial enzymes are specifically produced to be used commercially. They are typically found in products relating to pharmaceuticals, chemicals, biofuels, food, beverages, and other consumer-based fare. The advancement of biotechnology has allowed the manufacture of industrial biological catalysis through enzymes to increase exponentially. This is due to the flexibility of production, which traditional chemical processes can fail to match. If enzymes are immobilized for any reason, the interparticle mass transfer resistance can affect enzyme reaction rate. The applications of enzymes can be separated into three major categories: industrial enzymes, analytical enzymes, medical enzymes. Carbohydrates. Carbohydrates are made up of a significantly wide-ranged class of naturally occurring organic compounds, including sugars, starches, and celluloses. They are essential to the maintenance of plant and animal life. Carbohydrates are classified into three major groups: monosaccharides, oligosaccharides, polysaccharides. Monosaccharides are simple sugars, with the most basic form of carbohydrate units. Oligosaccharides contain two or more of these monosaccharide components, whereas polysaccharides have hundreds or thousands of them. Plant cell cultivations. Plants are a valuable source of various chemical compounds such as pharmaceuticals, flavors, pigments, fragrances, agrochemicals. These products are known as secondary metabolites. Their natural purpose is to act as a chemical interface between the plants and their surrounding environments, reacting to environmental stresses or defending against microorganisms. Despite substantial advances in chemistry, many secondary metabolic compounds are either too difficult or too costly to synthesize, so complex mixtures like rose oil cannot be easily manufactured. The majority of the plants that produce valuable substances grow in tropical and subtropical regions. As a result, these materials' availability and costs will depend on the political and economic circumstances of the native country. To produce secondary metabolic products from a plant, tissue can be cultivated in specialist conditions. The technical rationale for using plant tissue is based on the unique biochemical properties of plant cells. Cultivation of cells instead of the whole plant can provide several advantages, such as plant cells can be cultivated where they are needed regardless of weather and geographical conditions. The product quality can also be precisely controlled by eliminating the problems encountered in botanicals processing. Some metabolic products can be produced from the culture in higher quantities than would naturally occur.